Welcome to the last part of Area of Study 2. This video is about gene cloning. There are two other techniques that in a normal year we will learn about the bioethics, but this last kind of unit is on the application of those technologies that we learned about in Area of Study 2.1, so PCR, the gel electrophoresis, and the bacterial transformation. So normally we do DNA profiling and also genetic screening for babies, but they've cut those this year and we're only going to focus on gene cloning. So what does gene cloning mean? It's kind of what it sounds like on the tin. So gene cloning is about producing clones or exact copies of a gene of interest. So you get one gene and then you make lots and lots of copies of that gene. So this is very different to cloning whole organisms like Dolly the sheep. So this was in Unit 2. We did cloning whole organisms. We don't do cloning whole organisms in Unit 4, just about a particular gene. So I want you to think about the three techniques: gel electrophoresis, PCR, and bacterial transformation, and think about which one of those techniques in particular is going to be most applicable to gene cloning. And then we're going to go through two methods: in vivo, which is gene cloning, or what does it mean to be in vivo, and then in vitro, which means in glass, which is the more common way. Before we go there, I just need to come back to Unit 3 when we talked about the genetic code and these two words, but in particular, remember the genetic code is universal and it is also redundant. But in terms of this and this subtopic, when we talk about gene cloning, it's all got to do with the fact that the genetic code is universal. That means it's the same in us as it is in pigs, as it is in bacteria, that same genetic code. So it doesn't matter which organism produces the protein, the gene coding for it will always be the same. This is always the same. So you go, if the code is UCC, it will always code for serine. If the code is if the codon is GAU, will always code for aspartate. Yeah, it doesn't matter if that's in a human or another one. If it's UAG, it will always code for a stop codon. So that is really helpful because it means you can take genes from one organism and put them in another. So the first type of gene cloning I'm going to talk about is gene cloning in vivo, so in a living organism. So this is when you use a bacterium to produce many copies of a gene. So we've already done this in terms of the process. So if you need to, you can go back and watch the video on bacterial transformation. But in vivo gene cloning is just making many copies of a gene in a living organism. And the living organism we usually choose to do that in is a bacterium. So this is where we use restriction enzymes, cut out a gene, insert it into a plasmid, stick it together with ligase, and then we put those plasmids back in the bacteria and they do the cloning. They reproduce the plasmid, and so they are cloning that gene, making many, many copies of that gene. So that's the first type of gene cloning. The second type of gene cloning is in vitro or in glass or in a lab. And so this one, we're talking about PCR. So out of my three techniques, gene cloning can apply to bacterial transformation if we're talking about living things doing the gene cloning, or it can happen in glass or in a little test tube, and that is PCR. So PCR, what happens in PCR, just really quickly, you put in your DNA sample, your primers, your nucleotides, your Taq polymerase or your DNA polymerase, all of that goes into a tube, into your thermocycler, and then you start heating it up. Remember ABC, it goes high first, denaturing, splitting apart those strands. Then it goes cool, and we get the annealing of the primers onto each end. Then we go into the middle temperature around 72 degrees, and you get that extension. That the DNA polymerase starts moving down the chain. So this is that last stage here, and you get a new strand of DNA. Just before we finish about that, what are some of the applications? Why do you want to clone a bunch of genes? One of the biggest reasons that we want to do it is because we want to do some research into what the genes do. And so before you start doing research and experiments, you know we need to copy things because we need to be able to repeat the experiment. So whenever you want to research a gene and what that gene does, which is what gene expression means, that's why we might need to use either PCR or bacterial transformation to make many copies of it. The other thing that we need to do is we might have an application of gene cloning. So this one in particular, you would more likely use PCR. Yeah, and then you produce all of this funny data that's over here. You don't need to be able to understand what that is telling you, but it's just an example of some of the data that you can get out of the process of gene cloning. And then the second one is producing recombinant proteins or recombinant bacteria would be better. And we'll talk more about that when we talk about the applications in agriculture. So this is where you are doing bacterial transformation is one of those big ones there. And we use it a lot in agriculture for passing on traits and delivering genes and many copies of those genes to a plant so that it produces a particular protein that we're interested in. And that's all you need to know about gene cloning.